Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today we'll be talking to Karla Jarecke and Terry Loecke about methane fluxes in constructed wetlands. What purposes do constructed wetlands serve? What kinds of plant species live in them? How can these species and hydrology affect methane fluxes in these ecosystems? Answers to all these and more coming right up. I'm your host, Abby Morrison. Let's talk about science. Hi everyone, welcome to the show. Today we have Karla and Terry with us. Karla is a PhD student at Oregon State University in Corvallis, Oregon, where she studies soil hydrology and tree water stress in forest ecosystems. She received her master's degree in natural resources at the University of Nebraska, where her research focused on mechanisms of greenhouse gas emissions from wetland soils. She completed her bachelor's degree from Rockhurst University in Kansas City, Missouri, where she majored in biology and Spanish. Terry is an ecosystem ecologist and biogeochemist with the job titles of assistant professor and assistant scientist at the University of Kansas in the Environmental Studies program and the Kansas Biological Survey. He has several degrees, including a University of Northern Iowa bachelor's degree, his master's from Iowa State University, and his PhD from Michigan State University. He studies how land management, including agriculture and restoration, affects soil, air, and water quality. Hi, Karla. Hi, Terry. Welcome to the show. How are you both doing today?
Great. Thanks for having us on.
Doing well. Thanks for having us, Abby.
Wonderful. Glad to have you here. So today we're talking about wetlands and methane fluxes. So to get started, can you just tell me a little bit about constructed wetlands and wetlands in general, some of the functions that they serve, anything like that?
Yeah, I can start. So wetlands are really valuable on the landscape. They support a lot of ecosystem services, including biodiversity of plants and wildlife, and they aid in flood control, and they can filter and clean water by storing and recycling carbon and nutrients. And they're really critical ecosystems from a more global perspective because they remove carbon from the atmosphere and can store large amounts of carbon in the soil.
And then we're talking about hydrology and wetland plants as relates to wetlands today. Can you tell me a little bit about how those things interact and can affect each other? Maybe a little bit about the kind of methane life cycle as relates to plants?
Yeah, so the hydrology of wetlands can be, in some places can be very constant, persistent. Other places can be quite variable, and we might call all of these wetlands. Like an ocean is typically not thought of as a wetland in the terrestrial sense, but it is a wetland in some of the services that it also stores a lot of carbon when carbon falls into it. So that carbon can be stored long term, or one of the fates of that carbon is being converted to methane. And methane is a greenhouse gas. It circulates through the atmosphere. It has a fairly potent greenhouse gas, and that means it absorbs a lot of the heat that's being radiated off of the surface of the Earth. And so the methane is also a fairly long-lived gas, so once it's emitted, it stays in the atmosphere for a while. We have natural processes which will remove methane from the atmosphere. Some of those are related to ecosystems like wetlands that can oxidize or remove or consume that methane from the atmosphere. We also see this consumption in upland soils, so drier ecosystems have usually a large potential to remove methane from the atmosphere.
And to add a bit to that, you asked about the role of plants. So methane can it's produced in the soil, but it can be released to the atmosphere through plants. And the hydrology matters in that context because when the soils are flooded, the plants can potentially play a large role in how methane makes its way from the soil environment to the atmosphere. And this happens because the methane will get dissolved in the soil water, and the plants are actually taking that water up during transpiration. And so as it's making its way through the plant, it can actually diffuse out of that water and through the plant tissue and to the atmosphere.
And then plants can also add oxygen to the sediments which they're growing, and so the soils that they're growing, which can promote that oxidation. So there's a whole cycle, a whole series of feedbacks that can occur with plants, and methane, with hydrology being one of the main drivers of those cyclings.
Excellent. So with that background information, what hypothesis were you trying to test specifically in this experiment?
So we were interested to test whether plants and hydrology interact to control the amount of methane released from the soil to the atmosphere. And we did this by combining lab and field observations to understand if methane emissions differed from two common plant species at our study site. And this is northern water plantain and swamp milkweed. So we measured methane fluxes from these plants in the field at flooded area and in an area that was not flooded. And we took methane flux measurements in the lab in mesocosms that were flooded.
Excellent. Can you tell me a little bit more about these two different plants? What do they look like? How are they different from each other? That kind of thing?
Water plantain tends to be more erect. It stands up and so kind of looks like a grass that you might see. And both these plants can be upwards of several feet tall. The plantain, it has more of an erect type of posture. The swamp milkweed can have a nice flower on it. A lot of people value in wetlands for one of those indicator species that indicate a high quality wetland.
Wonderful. Yeah, I actually grew up next to a wetland, so I'm quite used to different grasses and little swamp wildlife, so this is close to home for me. So then speaking directly to kind of these two different phases, can you describe more specifically the different methods that you use? We'll start with just in the lab.
Sure. So in the lab, we started actually we needed to set up our lab experiment by going to the field to collect soil cores and seedlings of each of the plants. So we collected soil cores in the field by driving PVC into the ground, and this PVC is just PVC pipe that's about 15 centimeters in diameter and 30 centimeters long. And then we dug these cores out of the soil, and then we collected the seedlings of each plant species, 5 of each, and we brought them, all of this, back to the lab. And then we transplanted those plants into the soil cores. And the soil cores are inside these 5-gallon, like Home Depot buckets. And after giving them some time to recover from the stress of the transplanting, we flooded the mesocosm, or we added water to the bucket until the water was about 2 centimeters above the soil surface. And then once that was allowed to equilibrate for a couple days to create an anoxic environment, we put a plastic acrylic chamber that fit on the PVC and went around the plant. And this chamber had a lid at the top that had a vent and another port that allowed us to take a gas sample from the headspace. And we did this sampling once a week for 6 weeks to understand the methane flux from that soil plant chamber and see if they differed in chambers that had the different species. And we also had a control chamber set up that had no plants and just the flooded soils.
And are you cycling out the air that is in the chamber in any way, or is it just completely sealed for that entire time?
So these chambers are taken on and off. They'll be placed on over the plants for approximately an hour, and then allowed to, and then for the time in between sampling, they have no chambers on them whatsoever. So we're trying to make it so we're not changing the atmosphere a whole lot around the plants, so we're not letting them heat up to any great extent during this time. So the purpose is to sort of take a sample with the chamber and then get out and then come back and do it the next time.
Yeah, I like that. In my brain, I'm just imagining I'm thinking of like the glass jar that goes over the rose in Beauty and the Beast. And like, I know that that's not at all what it's like, but that's where my brain goes. But yeah, I really like that. But moving on, I guess, to the second phase of this, which was testing in the field. Can you tell me more about how you went about that stage of your experiment?
So in the field, we had a similar setup with the chambers, except this time we had a larger diameter PVC base that went around intact plants in the field. And we had these two sites, as I mentioned before, one that was flooded, and meaning it had standing water at the soil surface, and the other site was not flooded, so there was no visible standing water. And we established these chambers around 5 plants of each of the species, and we took the methane flux measurements daily for 3 consecutive days. So it was a very similar design in terms of the chambers, but the field experiment was a little bit shorter. It was 3 consecutive days versus in the lab we did this under flooded conditions for 6 weeks.
Why only 3 days?
It's just more challenging to be in the field, and it just takes more time, more logistics, and so we could only afford to do it for a short amount of time as sort of a test of the concept of what we're seeing. So
fascinating. So I have another question about your field experiment. So when you say that you're out in the field, what does that mean? Are you in like a constructed wetland that's just out in the wild somewhere, or are you at, say, your institution's specific constructed wetland? Is it a mix of different plants that you're trying to, it's like harder to isolate just one example of this kind of plant? What does that physically look like for you guys?
yeah, so I think that's really interesting because it matters a lot, and context matters on where you're at. And context matters to the people who might be using this information. Are there wetlands similar to the wetlands that we're studying here? So, in this particular field was slated to actually become a landfill. It was a corn and soybean field, and the municipality that we were in was interested in putting a landfill on this property because wetlands are tightly sealed up. They don't let a lot of water through because their soils are quite rich in clay. And so this one, the Parks Department, which we have to give a shout out to Five Rivers Metro Parks in the Dayton area, they purchased this land and then proposed to turn it into a wetland for the purpose of wetland mitigation acres.
And when you say Dayton, you mean Dayton, Ohio, correct?
That's right. And so they began to propose the idea of building a wetland, and they had presented this idea at the Wright State University that my partner and I were at the time. And my partner, Dr. Amy Bergen, picked up on this and said, oh, that would be cool to study a wetland. So we partnered with them to convert this cornfield back into a wetland. And so maybe 100 years ago or so, farmers started to settle this area and began to try to drain areas like this in order to plant row crops like corn and alfalfa, it would have been back then. they were able to struggle for many decades, really. We can see air photos going way back to the early days of flight, showing farmers struggling in these fields to try to grow a crop. Some years it was just too wet, and they would put in more and more subterranean artificial drainage in these systems. And with that, they had sort of changed the wetland very slowly into a farm field over time. Now, it still made a good wetland. We only had to break the tile that had been installed, the subterranean drainage, and then add some borders around it in terms of a berm or a kind of like a levee to be able to raise the water level a little bit. And so this area was partly submerged throughout the year and partly dry. And we studied plants all across the sort of transect between the areas that would be wet and dry. And that's where we'd see these two species near the sort of areas that were sometimes wet and sometimes dry.
So were you doing your wet and dry experiments at different times of the year then, or were you just kind of finding different areas within the wetland for those 3 days?
So yeah, that's a good question. So during that time, Karla and Kane Silva had gone out and identified places in the wetland where both these species existed, and of those locations, some were wet that day when they identified them, and some were dry on the surface that day that they had found them. And so they compared them, throughout the next 3 days. And those 3 days, I believe, were dry. We didn't have any precipitation those days. Karla didn't have as much precipitation as she wanted all that summer. But those are the circumstances in which we found those species, and at previous times that year or earlier, 2 years earlier or something, usually every year those species both areas would have been flooded, but one area just dries faster than the other.
Sure. I guess you kind of lucked out then that you were able to get your dry data. I'm so fascinated by this. I love hearing about scientists in the field and just the different things that they have to work around, because it's like you're outside. You just can't control it. And I find that just infinitely fascinating. And I just commend all researchers on just the cool hacks and workarounds that they find to do their work. Is just so delightful to hear about. Hi everyone, I hope you're enjoying the show. Interested in learning more? Karla and Terry's article, Plant Species and Hydrology as Controls on Constructed Wetland Methane Fluxes, published in the Soil Science Society of America journal, will be freely available for the next 2 weeks. You can find a link to it in our show notes. Let's get back to the show. I guess I would like to know next about the results. So starting again with the lab experiment, what did you find?
So in the lab, we found that the mesocosms that had the swamp milkweed produced about 8 times as much methane as the mesocosms with the water plantain, and about 60 times as much methane than the mesocosms without the plants. And then we also measured the CO2 in the headspace of those mesocosms and found that CO2 was dropping, or it was dropping in the mesocosms with the plants. So that was expected because the plants are taking CO2 out of the atmosphere. But we were excited to see that there was this difference between the two species, and that sparked our interest to go out to the field to see if we could see the same results in the field.
So just for anyone who doesn't know the term, what is a mesocosm?
It's basically our attempt to simplify a real ecosystem, take out a small piece of it and study it somewhat in isolation. So we try to mimic as much as possible the natural conditions of the ecosystem, but Meso means kind of middle-sized. And so these are middle-sized or smaller versions of the real deal. This is sometimes a successful approach and sometimes not. You don't really know until you try.
That is just the motto of science. So did the soil mesocosm have any fluctuations in the methane production, or was that just pretty steady? Like, was there any indication there were other factors feeding into that?
So the mesocosms that we created in the lab, there was no indication of any methane flux from the mesocosms that just had the flooded soils. There might have been a very small amount of methane, but I don't recall there being anything from the chambers without the plants.
Yeah, it was definitely very much diminished compared to the system with the plants. And that just goes to the dynamic role of plants here. They're feeding the microbes in the sediment, which the ones who are then using some of that carbon to produce some of that sugar, which the plants are secreting into the soil to produce methane. And without that, really the soil acts like it's, at least in these short-term experiments, the soil will act like it's starved of carbon. It doesn't have the resources it needs to be able to produce methane.
Fascinating. Thank you for that. So then what were your results in the field?
So in the field, we found that at our saturated site with the standing water, we had a similar amount of methane from the mesocosms with the swamp milkweed and the water plantain. And at the unsaturated site where there was no standing water, there was a small amount of methane produced from mesocosms with the swamp milkweed, but the mesocosms with the water plantain actually acted as a net sink for methane. So we didn't find any strong statistical evidence at that site for differences in methane flux between the species. However, the site itself, like testing whether or not there are differences in methane flux from sites that are saturated versus unsaturated, we found that there were differences, that sites that are saturated are producing significantly higher amounts of methane, which is what we expected to see. But we also expected that at that flooded site, there could be differences in flux, methane flux between the two plant species, but we did not find that.
Yeah, I think that's a really cool part of your project is just it's always so fun to find that there's more to discover, like obviously there's something going on there that is different from lab conditions to the actual field. So I'm sure you guys have put a great deal of thought into what that might be. So do you have any working theories on to what might have caused the change there?
So one thing I'll mention off the bat is that in the lab experiment, we actually had that stable water table. As I mentioned before, the lab experiment was much longer and we flooded the soils for about 6 weeks. So that set of conditions alone could have created differences in how these two plant species are affecting microbial methane production as well as microbial methane consumption, which Terry mentioned earlier is something that can happen because plants are actually moving oxygen from the atmosphere through the roots and into the soil. And that introduction of oxygen can actually facilitate methane consumption. So that's one guess, is that in the field we had a more hydrologically dynamic system where we didn't have the steady flooded conditions persist for 6 weeks prior to those measurements.
And we know that these plants, as they develop in the field, they respond to this dynamic hydrology and in ways that aren't that well understood. Some of them will produce certain types of tissues under dynamic hydrology that they wouldn't under continuously flooded or continuously dry conditions. And so there's a lot really to figure out about what these plants do in the wild. So we're still trying to understand, if you're a manager trying to use these species in a wetland, in a restoration or a reconstruction of a wetland, where would you put them in the wetland? What kind of rate would you put them in relative to other species and the seeding rate? And so there's definitely a lot yet to learn there.
Yeah, what a delightful mystery to get to research. It's so interesting to even think about the idea of like seeding a wetland. It's just an interesting picture to have to imagine. So obviously we've talked a little bit about some areas for growth in this research, but what are some areas for future research with this, either specifically with continuing the line of reasoning or questioning from this project, or just constructed wetland and hydrology research in general?
I think a cool follow-up study to this specific project would be to understand how differences in plant-specific methane fluxes could occur under varying levels of soil saturation. So you could imagine maybe repeating that field experiment, but under different, maybe prolonged water table scenario, under more dynamic scenario, and just kind of extend the life of the experiment to get more information.
Yeah, one of the things we're trying to do there is try to understand the degree to which wetlands are both supportive of the ecosystem services that we'd like to see in clean water, but also how are they contributing or mitigating the greenhouse gas effect that other parts of our systems are now exacerbating. So really one of the big questions is, we've been restoring wetlands and protecting wetlands for water quality reasons. Is it a good idea to restore them for air quality and climate change reasons? So all those fit together in sort of the policy. We might try to inform policy by thinking about the degree to which wetlands are good for those two types of, you think of the Clean Air and the Clean Water Act. Are they butting heads, or are they working together? So we want to inform policy, but we also want to give tools to managers of these systems so that they can do the best to meet the requirements of biodiversity, meet the demands of flood control and wildlife habitat, as well as hopefully start to ring the ear about climate change and how these wetlands interact in that system.
Yeah, and they really are beautiful systems, speaking of things like wildlife and biodiversity. Karla, in your time out in the field, were there any like fun animals that you got to see, or just, I always love hearing like field stories. So is there anything fun about that you'd like to share?
Well, I spent a few nights at the field site waiting for it to rain because my project, I did my master's project there, and I was basically waiting around for these large storm events to occur so that I could take gas samples from the soils before and after these big events to see how that might be playing a role in the mechanisms by which gas is transported from soils to the atmosphere. And I did often hear a pack of coyotes. Never saw them, but that was probably the most exciting I got. There were a lot of different bird species. I am not a birder, but I did run into several birders that would frequent the site and were logging their bird counts on eBird. So that was cool to talk to them about the different bird species. I can't remember them right now because it was 4 years ago, but there were a lot of birds, which was a cool aspect of how quickly, the introduction of wetland plants can really transform wildlife visitation.
Yeah, that's super awesome. I I'm a big fan of birds, big fan of wildlife. I was not expecting coyotes to come up as like your first thing, so that's fun. But yeah, they're just beautiful, beautiful areas. So for anyone out there who has not recently visited a wetland, highly encourage you to do so if you get the chance. But that also kind of brings up the next question, which is just really like, how is this research for people who are not researchers and, you know, don't have that background in hydrology and methane and the numbers side of things with this? Like, why is this research so important to just the average person on the street? And what are some key takeaways of why they should take heed of this kind of research?
So wetlands, they provide a bounty of ecosystem services: clean water, clean air, flood abatement, wildlife habitat. Managing wetlands for clean water, air, and biodiversity is an emerging interactive challenge that needs our attention. You know, these, as we said, are beautiful ecosystems to enjoy. And one of our goals is to make the less visible aspects of the qualities of wetland, like clean water and clean air, more visible, more intriguing to people who've been in wetlands and have really enjoyed the plants and animals, the more charismatic aspects of a wetland, because these are really important aspects as well. You know, it's hard to tell if water is dirty, and it's even harder to tell if the air is clean with your own eyes. So one of our goals is to make these more visible. Wonderful.
That's a wonderful cause. So thank you for dedicating your lives to doing this research. So then, now that we have people hooked on why this is so important, where else can they go for more information on this topic?
Yeah, that's a really multifaceted question. For landowners, people who might be interested in participating in programs to restore more wetlands, the U.S. Department of Agriculture has several programs associated with the 2015 Farm Bill and the most recent Farm Bill and the 2015 Clean Water rulings of part of WOTUS. And so there's programs like wetland mitigation as component of the conservation compliance program. I guess those are the most important ones that I can think of for land managers. For individuals, there's lots of groups who are very much invested in the quality and quantity of wetlands. Audubon Society, like we talked about, bird watching is an important one. Getting involved with your local Audubon Society or Ducks Unlimited or the Nature Conservancy are really important places to not only say, you know, yes, we need to protect and have wetlands, but how do you get involved? Your hands dirty and actually going out and volunteering and planting plants and clearing up trash if need be. Those kind of volunteer opportunities are abundant through Audubon Society, Ducks Unlimited, the Nature Conservancy.
Are there other ways for listeners to be involved?
Well, always voting your passions and your interests are a key component. We need support more than ever for legislators who are willing to protect natural ecosystems, restore the ecosystem functions that we all rely on.
And one other thing I would add is checking out if you live near a university or state school or community college, there could be citizen science projects going on there. And that's a really cool way to be directly involved in the science and help collect data. So citizen science projects are emerging as a huge database for scientists to collect more data because we are data hungry, and it always helps to have more data across space and time. And so that could be a potential way to get involved in the wetland science itself.
Yeah, that's great advice. Citizen science projects are super awesome, and I'm really glad that they exist. So yeah, listeners can check those out. We'll include links to those resources in our show notes. And then final question for you here is, what is one fun fact that people would not know about you if all they had was your research?
So my fun fact is that, I'm no longer measuring like anaerobic microbial respiration in the field or the lab. I am now taking this on at home by starting fermentation projects, including making beer and fermenting cider. So I'm in Oregon, where that's easy to get resources on how to do that. So that's something I've been doing for the last 3 years, and it's been really fun.
And I've been spending my time chasing my 4-year-old twins around the last few years. And so if you don't know that about me, that's how, if I'm missing, I'm usually with them.
How delightful. Those both sound like wonderful activities. So I'm so happy for you both. Thank you so much for being on the show. We have learned a lot. It has been a blast. And yeah, just thanks for your expertise and your research.
Thanks for having us.
Thanks for having us, Abby. And I also want to mention thank you to the USDA for funding part of this research, as well as the National Science Foundation.
Excellent. Thank you to them as well. Thank you for listening to Field Lab Earth. You'll find a link to today's paper and other resources for this episode in our show notes or on our website. If you have any questions, comments, or recommendations for show topics, please contact us at podcast@sciencesocieties.org or on Twitter @FieldLabEarth. If you'd like to hear more content like this, please subscribe, and don't forget to rate and review us on Apple Podcasts or anywhere else you find your podcast if you like our show. This podcast is a joint production of the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. Special thanks to Loecke for the use of their song Spook Castle on the intro and outro of our show. Opinions and conclusions expressed by authors are their own and are not considered as those of the American Society of Agronomy, Crop Science Society of America, Soil Science Society of America, its staff, its members, or its advertisers.